There's something they've never told you about vision. It's not what you see that defines your reality, it's what your brain decides to show you. The human eye isn't a camera. It doesn't record complete images, capture moments, or transmit an objective reality. What you experience as seeing the world is, for the most part, an active reconstruction, a constant prediction that your nervous system generates from incomplete fragments of information. And that changes everything you thought you knew about how you perceive reality. To understand why, we have to go back to the beginning, the anatomical beginning, the evolutionary beginning, the physical beginning. We must begin with a structure that is barely 24 millimeters in diameter, weighs less than 8 grams, and contains more than 130 million light-sensitive cells. A structure that takes about 10 years to reach full development, but which begins to function weeks before birth. A structure that, under ideal conditions, can detect a candle flame 48 kilometers away. The human eye. When you observe an object, what actually occurs is a chain of physical, chemical, and electrical events of a speed and precision difficult to comprehend. It begins with light. Light enters the eye through the cornea, a transparent dome that covers the front of the eyeball. The cornea is not simply a window. It is the first and most powerful lens of the visual system. It provides approximately 2/3 of the eye's total focusing power, around 43 diopters. Without the cornea, the eye could not focus on anything, and yet it is avascular, containing no blood vessels. It is nourished by the oxygen dissolved in tears and the aqueous humor that surrounds its inner surface. It is one of the few structures in the human body that can receive a transplant from a donor without requiring blood type compatibility, precisely because it lacks circulation. Behind the cornea is a chamber filled with a clear fluid called aqueous humor. This fluid is not static. It is constantly produced in the ciliary body, circulates within the eye, and drains through a structure called the trabecular meshwork. It maintains intraocular pressure, nourishes the cornea and lens, and acts as a shock absorber. When its drainage becomes obstructed, the pressure inside the eye increases. And this silent increase is what we know as glaucoma, one of the leading causes of irreversible blindness worldwide. In the center of this anterior chamber is the iris. The iris is the colored part of the eye, and its color does not depend on a specific pigment, but rather on the amount and distribution of melanin in its layers. Brown eyes have a high concentration of melanin. Blue eyes do not have blue pigment. They have so little melanin that light is scattered in a way that makes the tissue appear blue. The same phenomenon that makes the sky blue. The iris is not merely decorative. It is a muscular diaphragm of extraordinary precision. It contains 2 groups of muscles: the pupillary sphincter, which constricts the pupil in bright light, and the dilator muscle, which dilates it in darkness. This adjustment can occur in less than a second, changing the pupil's diameter from approximately 2 millimeters in bright sunlight to nearly 8 millimeters in total darkness. This is the difference between letting in 1 amount of light and letting in 16 times that amount. Behind the iris is the lens. Here begins one of the most sophisticated mechanisms in all of biological optics. The lens is a completely transparent, biconvex structure made up of specialized cells called lens fibers, which at some point in their development lose their nucleus and most of their organelles to become optically transparent. This transparency comes at a cost. Lens cells do not regenerate. From birth to death, the same cells remain, slowly compressing toward the nucleus over the years. This is why, with age, the lens becomes less flexible, less able to change its shape to focus on near objects. This is called presbyopia, and it is not a disease. It is the direct consequence of living with a system that was designed to last, but not indefinitely. The change in shape of the lens is called accommodation. When you look at something close, the ciliary muscle surrounding the lens contracts. The zonular fibers that support it relax, and the lens becomes more convex, more curved, more powerful. When you look at something far away, that muscle relaxes, the fibers tighten, and the lens flattens. This adaptation occurs automatically, in milliseconds, with a precision that no artificial optical system has been able to fully replicate. It is an active autofocus system, a biological biomechanism. The light, already focused by the cornea and adjusted by the lens, now passes through the vitreous humor, a transparent, gelatinous mass that occupies approximately 80% of the eye's volume. The vitreous humor is composed of 99% water, with collagen and hyaluronic acid giving it its consistency. It is biologically inert and, unlike the aqueous humor, does not regenerate. What is there at birth remains there for life. Over time, the collagen fibers of the vitreous can detach and float. And those shadows you sometimes see moving in your field of vision, those translucent dots and filaments, are exactly that. Fragments of your own eye's internal structure projected onto the retina. The retina. This is where light ceases to be physical and begins to be biological. The retina is an extension of the central nervous system. It is not just any tissue, it is brain tissue displaced to the eye during embryonic development. It has 10 distinct layers, measures a mere 0.5 millimeters in thickness, and contains approximately 126 million photoreceptors. There are two types, rods and cones. There are about 120 million rods. They are distributed throughout the retina, except in its center, and are exquisitely sensitive to light, even capable of detecting a single photon. They are responsible for vision in low light conditions, but they do not distinguish colors. There are about 6 million cones, and the vast majority are concentrated in a central region of the retina called the fovea. Cones require more light to function, but they are what allow for color vision and the perception of fine details. There are 3 types of cones, each sensitive to a different wavelength. S cones, sensitive to blue-violet, M cones, sensitive to green, and L cones, sensitive to orange-red. The brain compares the signals from these 3 types of cones and constructs the perception of color. This means that color doesn't exist in the physical world as you perceive it. What exists are electromagnetic waves with different wavelengths. Color is an interpretation, a neurological convention. And evidence of this is that a small proportion of people, mainly men, have a mutation that affects one of the types of cones, altering their relative perception of certain colors. They don't see the world incorrectly, they see it differently. And an even smaller proportion of people, mostly women, may have 4 types of cones instead of 3, potentially allowing them to discriminate a much wider range of shades. Research continues to explore this phenomenon. The process by which light activates photoreceptors is called phototransduction. It is a chemical cascade that occurs in microseconds. Inside each rod and each cone is a visual pigment made up of a protein called opsin and a molecule derived from vitamin A called retinol. When a photon of light strikes retinol, it changes its molecular shape, shifting from an 11-cis configuration to an all-trans configuration. This change in shape activates opsin, which in turn activates a G protein called transducin. Transducin activates a phosphodiesterase that degrades a molecular messenger called cyclic GMP. The drop in the levels of this messenger closes ion channels in the photoreceptor membrane. Sodium and calcium ions no longer enter. The cell hyperpolarizes, and this hyperpolarization is the electrical signal that travels to the brain. All of this, from the photon to the electrical signal, occurs in less than a millisecond. But the retina doesn't simply transmit this signal to the brain, it processes it. Bipolar cells, ganglion cells, horizontal cells, and amacrine cells form circuits that detect contrasts, edges, movement, and changes in intensity. The retina filters, compares, and amplifies. Before the information even reaches the brain, it has already been processed in ways that researchers are still deciphering. The optic nerve, made up of the axons of ganglion cells, carries these signals to the brain. It contains approximately 1 million nerve fibers, and at the point where the optic nerve exits the eye, there are no photoreceptors. There is a blind spot, a place in your visual field where you literally see nothing, which your brain automatically fills in with surrounding information, so efficiently that you never notice it unless you look for it. The signals travel along the optic nerve to the optic chiasm, where some of the fibers cross to the opposite side of the brain, and from there to the lateral geniculate nucleus of the thalamus, and finally to the primary visual cortex in the occipital lobe. But the process doesn't end there. The visual cortex is organized into specialized areas that process different attributes of what you see: color, movement, shape, depth, and facial recognition. There are dozens of interconnected visual areas that work in parallel and serially. Processing a complex visual scene can involve more than half of the cerebral cortex. And here lies the central point, the one that changes the perspective from which we look outward and inward. Most of the time, the brain doesn't wait for visual information to construct an image. It predicts it. It generates an internal model of the world based on past experiences, expectations, and context, and then compares the incoming visual signal with that prediction. If they match, the model is maintained. If there's a discrepancy, it's adjusted. Optical illusions are the result of this prediction failing. They are moments when the brain clings to an incorrect interpretation because it's statistically the most likely. What you perceive as seeing is largely the result of this predictive process, not a passive transmission of reality. There's yet another layer that makes this system even more extraordinary. The eye is never still. Even when you fix your gaze on a single point, your eyes make tiny involuntary movements called microsaccades, which constantly update the image on the fovea and prevent the photoreceptors from adapting and ceasing to respond. If these movements are artificially eliminated in controlled experiments, the image fades within seconds. It's not that the image disappears from the world. It's that the brain stops updating its prediction because there's no signal of change to warrant the update. Seeing requires movement. The eyes are also protected by structures that are often overlooked. The eyelids blink between 15 and 20 times per minute, distributing the tear film, a three-layered structure: a mucus layer, an aqueous layer, and a lipid layer, which lubricates, protects, and nourishes the ocular surface. The lipid layer, produced by the meibomian glands embedded in the eyelid margins, reduces tear film evaporation. When these glands are not functioning properly, dry eye is not only uncomfortable, but also directly compromises the optical quality of the system, because the regularity of the corneal surface depends on a stable tear film. Eyelashes are not merely aesthetic. They act as mechanical sensors that trigger a blink reflex before an object touches the eye, and as a physical barrier that deflects dust and debris from the environment. Eyebrows redirect sweat and rain away from the eyes. There is evolutionary engineering behind every structure, no matter how small or cosmetic it may seem. What we know about the eye has changed profoundly in recent decades. The understanding of phototransduction, which earned George Wald and his colleagues the Nobel Prize in Physiology or Medicine in 1967, opened the door to treatments for inherited retinal diseases. Gene therapy for forms of congenital blindness caused by mutations in the RPE65 gene is already a clinical reality in some countries. Electronic retinal implants, though still limited, have restored some degree of visual perception to people with advanced retinitis pigmentosa. Stem cell research is exploring whether lost photoreceptor cells could be regenerated or replaced. There are still no definitive answers. Clinical trials continue. The boundaries shift with each publication. What remains is awestruck humility at what already exists. In 24 millimeters of living tissue, there is an optical system without mechanical joints, brain tissue capable of detecting a single photon, a processor that reconstructs reality in real time, and an interface between the light of the universe and the subjective experience of the observer. No human technology combines all these functions in that volume, with that energy expenditure, with that adaptability, with that durability. When you look at the sky at dawn, or at the face of someone you know, or at a page of text, what happens is not that light enters and produces an image. What happens is that the physical universe, through electromagnetic waves, sets in motion a molecular cascade in specialized cells that migrate from the brain during embryonic development. And that cascade, amplified and processed by dozens of layers of neurons, reaches a cortex that constructs a coherent story about what lies outside. Seeing is not receiving reality. Seeing is interpreting it. And that silently happens within you every second you are awake, effortlessly, without instruction, as if it had always been the most natural thing in the world. What you have before you each time you open your eyes is not a window to the world. It is a biological masterpiece built in real time, layer by layer, neuron by neuron, over millions of years of evolution. The human eye is not perfect, but it is extraordinary, and understanding it forever changes the way you relate to what you see. If this journey through the hidden architecture of your vision has taught you something new, something you didn't know this morning, please like this video. That small action tells the universe that knowledge matters. And if you want to continue discovering the secrets your own body holds, subscribe to Revealed Anatomy. Each episode is a journey into your inner self. Sorry,